It's a huge pleasure for me to welcome Professor Paxton this evening. Robert graduated with a first-class honours degree in biology at the University of Sussex, and after completing his PhD at Sussex, he came over to Cardiff to join the Bee Research Unit with myself and John Free. After doing a brilliant job in Cardiff, Robert went on to study at Uppsala, in Sweden, at Tübingen, and then at Queen's University in Belfast. He is currently Professor of General Zoology at the Martin Luther University in Halle-Wittenberg in Germany. So, willkommen, Robert, and it's lovely to see you again.
Vielen Dank, Robert. Thank you very much for the very kind and generous introduction. What do we, if I can, what do we mean by pathogen spillover? Here I've taken a couple of examples. Pathogen spillover is the movement of a pathogen. Like you can see here on the left, I think these little dots are meant to represent viruses. The movement of a pathogen from one species to another, that may be man, which may or may not then go on to cause an epidemic within the human population. A couple of good examples of pathogens that spill over from livestock or wildlife into humans are anthrax, bacterium, which thankfully is not very common nowadays, and Ebola, which spills over from bats, possibly through intermediate hosts or directly into humans, and can then be transmitted onwards within human population. So pathogen spillover is movement of pathogen from its what's considered a reservoir host. It's the host in which it's found commonly, to a second or another host species in which it hasn't or is rarely found. I think for us humans, SARS-CoV-2 is a wonderful example. We understand how important pathogen spillover can be for the host, or in this case, for us humans. In the case of SARS-CoV-2, in this lower example, probably the virus originated in a bat, and probably went through an intermediate host, for example, the pangolin, probably maybe another intermediate host before entering the human population. Now this, I think this diagram from this paper from 2020 gives the human as the final host. It wasn't, that's rather arrogant, I think, in some ways, or premature to state that, in that we now know that this is a diagram showing the different variants of Omicron that arose from 2020 through to 2022. And here on the y-axis, we have the number of mutations on the spike protein. So there's a genetic difference from its origin. And you can see that a number of variants have arisen. In the pink here, we've got alpha, and then in green, we've got delta arising, which is a little bit more different again, further from the origin. And then more recently, we've had Omicron last year, since 2020, which itself is spreading now. But that's not the end story for Omicron, because that, we're still going. Yes. Here is a phylogeny of SARS-CoV-2. Looks like a very complicated phylogeny. But what it shows is that from humans, and most of these genomes, these gray bars here, have been taken from humans. In some cases, the virus has gone on to infect dog, domestic cat. Tell me, please. What am I looking for? I'm looking for the animal that's used in the fur industry. I've forgotten the name. I've gone blank. I'm sorry to say. So let me just show you then here. Tell me, Rob.
Ferret.
Ferret. That's it. Okay, ferrets and an equivalent. Thank you very much. This is the species name of the ferret. Thank you. And we can see that actually those variants that are now found in dog, or have been found in dog and cat and ferret, but also in white-tailed deer in North America and felines, they are derived from the human. They're slightly different from the corona that's found in humans and derived from it. So it's clear that SARS-CoV-2 is spilling over now from humans to our domestic animals and even to wild animals from an origin in humans. And this is based upon whole genome sequences. So, but these viruses and other pathogens, they don't just spill over from wild animals and from domestic animals into humans. They also spill from domestic animals, and as humans, you see in the last example, back into wild animals. And I'd like to give an example here, which may be analogous to what's happening amongst bee communities. So in the late 19th century, zebu cattle were brought from the Indian subcontinent to Northeast Africa, part of colonialism. They were brought in, they were carrying with them rinderpest, a deadly virus of cattle and other ungulates, unbeknown to those that brought in the zebu cattle, that are somewhat tolerant to rinderpest. The disease rinderpest spread rapidly through Africa, south of the Sahara, leading to over 95% of cattle, for example, here in South Africa, being eliminated in a short space of time. Thankfully, rinderpest has now been eliminated from the world by the United Nations, FAO, and World Health Organization. But the important point here is that when the cattle got infected, not only did they infect other cattle, but they also infected untold numbers of giraffe, zebra, gnu, and other ungulates in Africa, and probably changed the ecology of the environment, sub-Saharan Africa. So they had a major impact. And here we've got an example of, although not quantified, but certainly qualitatively good descriptions of wildlife being eliminated, other ungulates being eliminated, and other herbivores being eliminated, mammalian herbivores being eliminated from sub-Saharan Africa, with major ecological consequences, also for human health ultimately, but not directly. Okay, so the same may be happening in bees, or at least the suggestion has been that there may be spillover. So there is transmission between honeybees of pathogens, which may spill over presumably at flowers, but not really known, to wild bees species, which may cause their decline. And it's this hypothesis that I've looked at in some of the research, part of the research agenda that I've had in the last 10 years, 12 years at Halle in Germany. And it's that I want to explain to you today, or at least present to you some of the data that we've generated. Really looking at three questions. Does spillover occur amongst bee species? How does it happen, or where does it happen? And thirdly, does it have an impact? Is it a problem? And I'll have a series of little questions that I'll pose going through this talk, which will give me the opportunity to tell you something about the research that we've done. When I present research which is not part of what we've been doing, hopefully I'll remember to tell you clearly about that. So does spillover occur amongst communities of bees? I think there's pretty good evidence that the answer to that is yes. I think the best evidence is data that we have collected. This is Mark Brown, Matthias First, and other members of my group, who is at Royal Holloway, and other members of my group. And this is, I think, the best evidence that there is ongoing spillover. We've collected in 2011 from 26 sites in Great Britain, honeybees and 2 species of bumblebee from each of these 26 sites. So we have gone to the site and collected from flowers in a defined area, in a defined period of time. 20 honeybees, 20 of the commonest bumblebee species, and 20 of the second commonest bumblebee species. And these are the locations where we've collected. The different colors represent the different bumblebee species that were commonest or second commonest at those sites. Needless to say, large number of bees are collected. And then we screen them for viruses and microsporidia. And in particular, I'm going to tell you about deformed wing virus. The screening revealed this distribution of virus in honeybees and bumblebees. On the left, we have a heat map showing where the honeybees that were collected, this is collected from flowers, were infected with deformed wing virus. And you can see the heat map shows that in, for example, this must be around Cambridge or so in East Anglia, or just before East Anglia, up to 80% of the honeybees were infected. And down in Cornwall and Devon as well, 60 to 80% of the honeybees that we collected, this is present in Apis, were infected. We get almost the same pattern, statistically a similar pattern in bumblebees. In the same locations where there was a lot of virus in, or many of the honeybees were infected with deformed wing virus, so were many of the bumblebees in East Anglia in this case, and into Essex, and in Devon and Cornwall. Admittedly, the prevalence is lower, so only up to 25% of the bumblebees were infected, suggesting that the honeybee is the reservoir host of this virus, and that it presumably is spilling over, or could be spilling over, into bumblebees. Clinching evidence comes from sequencing. We've sequenced the viral variants in these locations, in fact, from 4 locations, and generated a phylogeny of the virus. This is deformed wing virus. So this is a phylogeny of the virus. And those virus variants that we obtained from bumblebees are in red, and from honeybees in black. And you'll see at this location, which is probably L here, which is in Essex, the viral variant in bumblebees is identical to the viral variant in honeybees. At this location in Cornwall, the viral variant that we picked up in honeybees is identical to the viral variant we picked up in bumblebees at that location. But both are slightly different from the variant in Essex here. So that means if they're sharing virus, they must be exchanging it. But from one geographic location to the other, it's a slightly different variant that's present. They must be sharing the viral variant on a daily basis, or at least within the seed collected. So this is an ongoing, this is evidence for ongoing sharing. Doesn't necessarily show which direction the virus is going in, but it's clear evidence of ongoing sharing. So spillover, movement of virus is certainly deforming virus. Movement of this pathogen is going back and forth, is happening amongst honeybees and bumblebees in GB, Great Britain. I'm going to go on to another experiment now and tell you some more recent work that we've just undertaken, looking at viral sharing and pathogen sharing in general amongst honeybees and bumblebees, focusing on bumblebees because that previous evidence in GB suggested that bumblebees might be recipients of virus from honeybees. So in Göttingen, now Göttingen is a city which is west of Halle by a couple of hours. It's more or less the geographic center of Germany, more or less. Right in the middle of Germany, surrounded by agricultural land. And at 16 sites, fairly distant from one another around Göttingen, PhD student working with me, Patricia Pluter, and I'd like to thank her at this point for her efforts in this project, along with colleagues from the Göttingen University, Katrin Westphal and her colleagues at Göttingen University, we have placed out 4 honeybee colonies and 2 bumblebee colonies at these 16 sites. They incidentally vary in the amount of organic agriculture they have, and flower strips that they have, and perennial natural, semi-natural habitat, which is the focus of the project. I'm not going to tell you about the relationships with those to see whether organic agriculture might benefit bees. It does actually. It seems to have a big effect. What I want to tell you about is the pathogens that we find in these honeybees and the bumblebees that we have placed at those sites. So these are not bees that we've collected randomly from the field, like in the first study. Here we're actually looking to see whether they share in real time when we have placed out the colonies. And this is the summary that I'm going to show you. At the top, you can see the prevalence of pathogens in honeybees, and at the bottom is the prevalence of those same pathogens in bumblebees. So we've got honeybees at the top, bumblebees at the bottom, and they were sampled twice, once in early summer and once in late summer. And what you can see is, okay, in the honeybees, they've got deforming virus type A, they've got deforming virus type B, quite a bit, sac brood virus, black queen cell virus, an awful lot. This is the prevalence here, over 75% to 75%, over 75% of them infected. Got acute bee paralysis virus in the honeybees. And then we have trypanosomes like Crithidia and Lotmaria, and Neogregarines apicystis, which is a bumblebee gut pathogen. You'll see in the bumblebees similar patterns, actually. So clearly, it's not just deforming virus that they're sharing. They're sharing, these bumblebees are sharing with honeybees a lot of virus, a lot of the viruses, and also other pathogens as well. So pathogen sharing and presumably pathogen spillover then is not restricted to deforming virus. It's reflected in a number of different pathogens and not just viruses. But now that's been focused on honeybees and bumblebees, I'm going to move on and say, well, okay, we've got the idea that honeybees may be a reservoir population, certainly for deforming virus. For the other pathogens, it's not clear. And they seem to be, at least for deforming virus, spilling over into bumblebees. The question is, what about other wild bees? We've got another, in Britain, we've got another 250 odd species of wild bee, and beyond the honeybee and the 30 or so species of bumblebee that we have. So what about other wild bees? So here I'm going to tell you about another data set now that we've recently collected within a project that it's got this wonderful name, Voodoo, Viral Coevolutionary Dynamics of Wild Domestic Pollination Global Change, which is funded through the EU, through their Biodiversa program and the German Research Council. And here we've got Halle, and in some 10 or so kilometers away or further, we have a series of sites from which we've been collecting honeybees, bumblebees, and other wild bees, and a large number of other wild bees, and examining them for their pathogens. So we've got 12 independent sites collecting using sweet nets. We collect from flowers, honeybees and bumblebees and wild bees, and we identify the wild bees, and then we check them for virus. And yeah, in this case, it's principally we've been looking at deformed wing virus and black queen cell virus, because these are the pathogens which are the commonest viral pathogens of the honeybee. And what we find here is, this is a summary of those data for those over 1,000 bees. We see black queen cell virus and deforming virus B in blue and green respectively, common in honeybees, apis here. So this is the prevalence here. Practically all the bees have got, honeybees have got black queen cell virus in them, and over half of them have got deformed wing virus B in them. This is for the honeybees. For the bumblebees, there are similar pattern, but lower prevalence, which reflects what's happening in Great Britain. For the other bee species, wild bees, Andrena species, Colites, Halictus, Lasioglossum, those are the larger ones where we've got large groups where we've got large numbers. You'll see occasionally they've got virus in. So at least from the German data, we can say that yes, there is seemingly that bumblebees seemingly have the same pathogens as honeybees, and probably there's spillover happening from honeybees to bumblebees, but it doesn't look as though, at least in our data sets, we often get those same pathogens in the other wild bee species. So maybe they're somewhat protected from, for whatever reason, from this pathogen spillover. I must say there's colleagues in Switzerland, Poland, and France that have done equivalent sampling. The Swiss data set looks similar to ours in Germany. Those in Poland and France suggest that actually quite a few wild bees are infected. So the prevalences of these pathogens is a bit higher there than it is here in Germany. Anyway, the evidence, the data that we have suggests bumblebees, yes. Other wild bee species, a little bit, maybe. And this is just summarizing here. Black queen cell virus, very prevalent in honeybees, somewhat prevalent in bumblebees, and hardly at all in wild bees. For DWV-A, this is which we don't find it hardly at all in Germany anymore, and DWV-B, variant B, genotype B, yes in honeybees, somewhat in bumblebees, and wild bees hardly at all. And I'd like to thank here Karima Bassett, who undertook her master's generating the data. So why do we focus on spillover of deformed wing virus? I think that's probably pretty clear to all of you why this virus in particular is important. You know, the last 10, 15, 17, 18 years, there's been a lot of attention paid to honeybee decline. First initiated, I think, at least beekeepers went to the journalists and politicians and complained about big losses. We think that varroa mites is the major cause of that, and the reason for the death is that the varroa mites transmit virus, in particular deformed wing virus, which leads to these crippled wings, and as we've shown in other research, rapidly causes the death of the bee that's infected. The varroa mite is exotic. It was introduced from East Asia to Europe and the Americas from Japan, and is now found more or less worldwide, including Australia. This is a lovely article by Kirsten Traynor that gives a nice history of the origin of and the spread of mite. It's restricted to the honeybee, and it is not found on wild bees, bumblebees, or other wild bee species. So, and the varroa mite, because it transmits viruses, in particular deformed wing virus, when it's feeding on pupae, or in, like here, where it's breeding, or when it's feeding on adults in winter, when it's overwintering with the adult bees in the winter cluster, then it transmits virus at the same time, particularly deformed wing virus. And now this is research not from my group, this is from Stephen Martin and his group. He showed very clearly, for example, that when varroa mites enter a population of honeybees, the prevalence of deformed wing virus increases rapidly. Here we've got islands of the Hawaiian Islands, when initially the honeybees were sampled before varroa mites were found. This is years of varroa exposure 0, 0, 0 in these years. Here, after 1 year of exposure, 2 years of introduction of the mite, or 3 years of introduction of the mite, we can see that deforming virus prevalence leads to every colony having deforming virus in it, which is probably the case everywhere where varroa mites are found. And also, here's the viral titers. The viral titers shoot up. So there's 10 to the 10s is 10 billion virus particles in a bee when varroa mites are present, of deformed wing virus particles or virions, genome equivalents in a bee when varroa is present. So that's why we focus on deformed wing virus, because honeybees have got a lot of deformed wing virus in them as a consequence of being infected by varroa mites. Okay, so pathogen transmission is happening. Pathogens seem to be moving between species. The suggestion is that it's going from honeybees to bumblebees, potentially also to other wild bee species. Where is it happening? Varroa mites only transmit it between honeybees. So how are the wild bees picking it up? The usual hypothesis is that it's on flowers. So to test that explicitly, we've undertaken a small study in which we haven't not been able to use genetically modified virus and see where it's moving, but we've used an indirect way of testing whether flowers, or providing evidence that flowers seem to be the place where the honeybee and the bumblebee may be sharing virus, and where this may be picking up virus. So here is a map with it's a very plain map with Halle in the middle. This is where I am right at this moment as I speak to you. And the yellow little pointers show you where we've sampled honeybees and bumblebees. And I'd like to thank Christine Hesse, a master's student, for undertaking this, this research. And she visited these 12 sites, collected 10 honeybees, 10 bumblebees, 10 of Bombus terrestris, the common bumblebee, 10 of Bombus lapidarius, the red-tailed bumblebee, also common, 10 of Bombus pascorum, the brown bumblebee, or the carder bumblebee, common carder bumblebee, all 3 common bumblebees. So these 4 species she collected at each site. And moreover, for one day at each site, she patrolled the area at which she'd collected these bumblebees and recorded continually which flower was being visited by which bee species of these 4 common bee species, honeybee and these 3 common bumblebee species. And using all this data, she collected hundreds and hundreds of records, and she could see to what extent honeybees were sharing flowers with Bombus terrestris or Bombus lapidarius and Bombus pascorum. So for each site, she could set up what we call an ecological network. So imagine these black and grey bars are composed of a series of lines, each line representing one record of a bee visit. Here we've got 276 honeybee visits here. In these, there's 276 lines here. And these black lines representing these 276 observations at one of the field sites show you which plant species on which flower it was visiting. It was visiting, so all of these here were visiting this plant species. A few were visiting this plant species. Many honeybees were visiting this plant species, Carduus anthocoides. Now, we've got the same for this bumblebee, Bombus lapidarius. And you'll see Bombus lapidarius is often visiting the same plants, flowers that honeybee is visiting. But Bombus pascorum is visiting a range of different flower species, largely from Apis mellifera. And we can quantify to what degree there is a so-called niche overlap between honeybee and these other 3 bee species. And this is what the niche overlap looks like. It's a value which ranges between 0 and 1. 1 means a lot of overlap, 0 means little overlap. And you can see the red-tailed bumblebee, Bombus lapidarius, has much greater overlap across those sites than does Bombus terrestris, and that has more than Bombus pascorum. Yeah, so you can see. So the hypothesis would then be Bombus lapidarius is most closely related to, or visiting the most similar, is most similar in its flower visitation to Apis mellifera. You'd expect it to. We expect that to be the most infected. The prevalence of virus that's in honeybees to be highest of this bumblebee and lowest in this bumblebee species here in Bombus pascorum. Let's have a look at the patterns, and indeed that's exactly what we find. Looking at the prevalence of a black queen cell virus, very common in bumblebees, very prevalent, we can see that there's a clear pattern that the species of bumblebee which is most similar in foraging has the highest prevalence of this virus of all the bumblebees. The Bombus terrestris intermediate and Bombus pascorum the lowest. Similar pattern here for deformed winged virus B. So if we look at the relationship between viral prevalence in bumblebees with their niche overlap with honeybees, there's a clear relationship there, suggesting that indeed it is at flowers, and if a bumblebee shares flowers, a lot of flowers with honeybees, they're likely to often be infected, and if they don't, then they're not likely to be infected. Again, suggesting that the virus is going from honeybee, the reservoir host, presumably, to bumblebees. Now, is transmission only in one direction? Well, we've undertaken some experiments to try and test that. It's difficult to do these sorts of experiments in the field. but we have done experiments in the laboratory to test for spillover, pathogen spillover, using deformed wing virus. We've taken honeybees, and here's me with a PhD student, Tabea. I'll mention her again in a minute. These are the hives that we use. This type of equipment, you might recognize it. BJ Sheriff should be happy that I still wear his bee suit that I bought, that I think Rob Pickard bought for me many years ago. It's still in use. But this is the short-lugged frames, very difficult to manipulate. This is German normal size. We have taken freshly emerged honeybees, and then we have injected them with virus, with our viral inocular, so that we have infected honeybees. And then we have taken bumblebees from commercial Bombus terrestris colonies that we bought, and we have injected them as well with virus. So using, and I'd like to thank here Anya Tehel, PhD student, or now finished, so now postdocing, Anya Tehel, former PhD student of the group, carrying on now with us still as a postdoc, who undertook these experiments as part of her PhD. So she then had infected bumblebees and infected honeybees. So she knew they were infected. She had injected them with the same amount of virus. And then she set up an experiment to see whether they would transmit the virus. Now, this looked like a little bit complicated. I'll try and walk you through this experiment. She introduced then into cages 8 honeybees that she had infected, young honeybees that she'd infected. And she added, so they are the donors, if you like. She then added 8 honeybees that were uninfected, also the same age, to see whether virus would be transmitted. This is a control. She then introduced 8 honeybees infected with virus as donors, with 8 bumblebees that were uninfected. These are in green. The idea is that they're uninfected, although the bumblebees are a little bit larger. Then she did the opposite. She added 8 bumblebees that were infected to a cage and added 8 honeybees that were uninfected. And then she added in other cages, she added 8 bumblebees that were infected with 8 bumblebees that were uninfected. And obviously there are controls as well. And this is a highly replicated, so she had many cages of these treatments. And then she looked after 7 days, after 14 days. First of all, she looked to see whether virus could be found in the recipient bees. Now, this is the experiment at the top, where there is full contact, if you like, between the bees within a cage. She replicated the experiment in which she kept the different species separated, or the recipients and the donors separated. And the only thing she did was to remove or to move the feeder, the sugar feeder of the bees in the cage, from the infected to the uninfected one, then give the infected one a clean feeder each day, every 24 hours, moving the feeder from the infected to the uninfected to see whether just using the same sugar source would lead to infection. And the data of this experiment reflect exactly what happened with the upper experiment. I'm going to just show you the results of the upper experiment. And here are the results. So when she mixed infected honeybees, Apis, with uninfected honeybees, Apis, she found the infected honeybees were obviously all infected. So all cages had honeybees, and the infected ones were infected, as you'd expect. We infected them, and of course they're full of virus. And the uninfected ones were also infected. Oh, and here you can see the titers that they had. So when honeybees are infected, when we infect honeybees, after 7 days, they got 10 to the 12. That's a million virus genome equivalents in them, virus particles in them, a hell of a lot. But the honeybees that we've mixed with them had 10 to the 10, 10 to the 9, 10 to the 10, 10 to the 11. So that's 10 to the 12, almost the same amount of virus in them. So they'd obviously picked up virus from their cage mates. There's the only way they could have picked up the virus, because controls had, in the control cages, We had no virus at all. So clearly virus is spilling over. It's being transmitted from honeybee to honeybee. Well, we know that's the case. That's just a control. Interestingly, the virus, when the honeybees were infected, they transmitted readily to bumblebees. So they also had a lower titer, but nonetheless transmission happened quite readily. Critical point here now, what happens when the bumblebees were infected and the honeybees were not? Here are the data. The bumblebees we infected were infected indeed, but the honeybees or the bumblebees, the uninfected bumblebees we mixed with them, were not. So bumblebees that we infected in pink had virus in them, in both cases. The honeybees that were uninfected that were introduced did not have virus in them. The bumblebees that were uninfected that we introduced with the bumblebees did not have virus infected in them. So it looks like the virus can transmit readily from honeybee to bumblebee. But at least in these laboratory experiments, it didn't go from bumblebee to honeybee. I suspect it's a matter of titer. What I will say is that if it is going to honeybees from bumblebees, it's at a much, much, much lower rate that it's being transmitted. So the virus is effectively principally going in our experiments from honeybees to bumblebees, which reinforces the observational data that we had and the phylogenies that we generated of the virus in that very first data set that I showed you. Right. What impact is the virus having then on the bumblebees in the laboratory? Again, we've got previous evidence. This is from our first et al. Paper, the first data set that I showed you, that included an experiment in which bumblebees were fed virus and then kept individually in isolation. And what was found is that those that were fed with a control solution lived 20 days. This is survival on the y-axis, so this is length of time after treatment. Control just fed sugar syrup live longer than, or have a higher survival than bees that are fed with than bumblebees that are fed with virus. So we thought, well, okay, they're kept in isolation. Let's try a little bit more realistic and not just use deforming virus, unknown genotype, but let's use a particular deforming virus type A, deforming virus type B, and black queen cell virus in the same sort of experiment, in which we either feed the virus to the bumblebees or we inject the virus into the bumblebees. And here we're feeding one of these viruses, and of course we've got control. And let's feed them. The idea was from Anya to feed them or inject them, and then see how quickly the bumblebees die. Now, so the bumblebees, they're fed or injected, put into cages like this, so they survive quite well in these little cages, given a continuous supply of sugar, and we check for mortality and then also quantify virus in them every so often. This is just a test of our inocular. The inocular we injected into honeybees, either DWV genotype A or type B or black queen cell virus, and you can see it kills the bees very quickly or quite quickly compared with a control solution, which is what you'd expect. You know, these are viruses from honeybees that we know when we inject them into honeybees, they kill honeybees very quick, adult honeybees very quickly or quite quickly, and that's exactly what we found. That means that our viruses are active. They're not being degraded. These are the viral titers that we get. So when we inject a virus in, we find a lot of that virus in the bee. So when we inject DWB, black queen cell virus in, we get a lot of black queen cell virus in the bee. So just everything's working fine. What happens though when we give the same viral variants, the same virus, DWVA, DWB, or black queen cell virus to bumblebees? When we feed them with bumblebee, we feed the virus to bumblebees, compared with control, no difference. Oh, the bumblebees that we feed do get the virus inside them. We feed them with black queen cell virus. They've got black queen cell virus in them. We feed them DWVA. They've got DWVA inside them. We feed them with DWB. They've got DWVB inside them, but it doesn't make any difference to their survival in the laboratory. When we inject the virus into them, which is not really a natural route of infection, it's likely to have greater impact. Again, no difference in survival. The correct virus is found, but no difference in survival. Very strange. So it looks like for us, though we know the virus is infective in honeybees and kills honeybees, these bumblebees seem to be quite tolerant to the virus, suggesting little impact. Only when we starve the bumblebee do we find, only when we starve the bumblebee, take away its food, and it's been injected with deforming virus A, do we see that there is some reduction in the longevity of the bee. So only under very stressful conditions. So that's in a laboratory experiment. And we thought, okay, laboratory experiment, well, what happens, out in the field where it's much more stressful? Maybe they're often in these sorts of situations where they're almost starving. So in this case, so here we've tried to look at the impact then of bumblebees in the field. And so the experiment goes like this. We take bumblebees from the laboratory, we infect them either by injection or by feeding. Then we introduce them to into colonies, which we've placed out in the field. And this is DWVA that we've been using here. And we individually mark the bumblebees and we see how long they live, and they quantify how much virus is inside them. And this is Tabea, again, a PhD student who has undertaken these experiments. Here you can see her adding bumblebees to infected bumblebees to field colonies. And what you can see, the results are that for the bumblebees that have been fed with virus, there's no difference in their survival compared with control bees, even in the field. Those that are injected with virus live slightly shorter than those that were injected with control solution, suggesting there is some impact in the field, but it's a small impact. Okay, I'm going to now take you over in my last data set to the Yucatan Peninsula, where we've tried to replicate that using tropical bees. So the Yucatan Peninsula here in Mexico has a number of stingless bee species that are native to the region. One of them in particular, Melipona beachy, is a particularly important species culturally and economically, was economically important. Here are the numbers of species of stingless bee in the tropics that you see in these different tropical regions. So Latin America is quite rich in stingless bee species, which are somewhat closely related to honeybees. In the Yucatan Peninsula itself, there's over 15 species of stingless bee, including this Melipona beachy. Melipona beachy itself is an iconic species maintained by in a traditional way that's been in the way it's been kept for the last 2,000 or more years. And these are ancient Maya codici, and one sees within them bees. For example, this is a bee, ancient Mayan symbol of a bee, and these are honeypots, stingless bee honeypots that are being collected by a beekeeper. So you can see there's a cultural relationship between and connection between the communities in the Yucatan Peninsula and this bee in particular, which is depicted here in these pictograms taken from the Codice Maya. So, important species. Does the honeybee transmit, or is this virus transmitted across to this species, to Melipona beachy, and does it matter? Well, certainly they're often visiting flowers. Here we've got same flowers. Here we've got a honeybee at the top and a Melipona beachy at the bottom, visiting the same flower. So potentially there could be spillover of pathogen. Is it happening, and does it make any difference? Fernando Fleitas, a PhD student currently with me, has been working on this problem. He's been collecting, he collected honeybees from field sites, and from the same field sites from Melipona boxes, collected Melipona bees, then tested them for virus. And what we find in terms of black queen cell virus, it's everywhere in the honeybees. These are Africanized honeybees in red. In blue, it's also found in Melipona beachy at the same sites where it's found in honeybees. Not always, but quite often. Much more abundant, much more prevalent in honeybees. Deformed wing virus A is a lower prevalence, but also occasionally found in Melipona beechii. And deformed wing virus B hardly found at all in honeybees, surprisingly for me. And we haven't yet found it in Melipona. So those are the prevalences. We can see lots suggesting honeybees are host for black queen cell virus and deformed wing virus A in these locations, and maybe Melipona beechii is a recipient. The question is, does it have any impact when they're infected? And I will just briefly tell you the experiment that he did. He fed, so Fernando fed Melipona bees with virus or control without virus, and then tested for the longevity in cages. So he put them back into little cages and looked to see how long they lived, either with virus, either DWVA, DWB, or black queen cell virus. And what he found is, indeed, they survived shorter. So the control is here in green, and the treatment groups in these other colors, red, purple, and blue, they died more quickly. Slightly more quickly, but more quickly. So this is a lab experiment, but unlike with the bumblebees, here with the stingless bees, they seem to be suffering somewhat from the virus, living shorter, dying quicker than control Melipona when fed virus, one of these three viruses from the honeybee, suggesting there may well be an impact in the field. We don't know what's actually happening in the field, and it's really important, I think, to run experiments there. But anyway, the suggestion is that it may be happening in this tropical area of Yucatan, tropical area of Mexico. There may be an impact. So, indeed, maybe we are suffering a similar fate. I don't think it's as dramatic as has been the case with rinderpest in cattle, but I think honeybees and potentially other wild and other bee species may be suffering a similar sort of fate, in that nowadays every honeybee colony that's infected with infested with varroa has black DWV virus in it, and unless varroa are controlled, this is going to be the consequence of lax beekeeping and no control in the vast majority of cases, with also potential consequences for wildlife and wild bee species. Okay, so yes, DWV is spilling over into wild bees. Yes, it does seem to have an impact, although we don't really know in the field whether it leads to population decline. And that's an open question that still needs to be addressed. Okay, at this point, I'd like to thank the funders, collaborators, the working group in Halle and previously at Queen's. And I'd like to thank you for your attention. And at this point, Rob, maybe I should end the slideshow. Is that good?
Yeah.
And stop sharing. Is that right? So I go onto camera.
Yeah.
Yep. And then you can see me and I can see you at last. And I can turn off the email, which I was just about to send you an email in case there's a breakdown in communication. And would you like me to answer some questions?
Yes, if you would, Rob.
Yeah, sure.
It's a really interesting subject, no doubt about it. I was going to ask you whether or not the bumblebees that had picked up the deformed wing virus actually showed the symptoms of deformed wings.
No, they don't, because that's a symptom that arises when a bee is infected as a pre-pupa or in its pupal stage. So, in our experiments, something like 60% of the bees that we infect as pre-pupae or as white-eyed pupae will end up with deformed wings. Not all of them will, and that's probably reflects what happens when varroa feeds on a honeybee. Yeah. Now, it's a good question you pose, though, actually, Rob, because there are often reports of bumblebees with deformed wings. and they often are packed jam full of deforming virus. So where are they getting it from? I mean, maybe it's brought back in pollen and fed to them. So it's an interesting. There's even a website in Germany for reporting bumblebees with crumpled or crippled wings, suggesting that it's actually quite widespread. I don't think all of those cases are due to deformed wing virus, but the first ever evidence that deformed wing virus was in bumblebees came from a group headed by Elke Genisch in Germany in 2006. And she discovered deforming virus in bumblebees by picking up bumblebees in an apiary that had crumpled wings. So there might well be some connection there, Rob. So good question.
Yeah, the one thing that did interest me was the discovery that 8% of the human genome is actually derived from viruses, that their evolutionary line has contracted over the long periods of time. I don't suppose we know anything about how much of the honeybee genome has been derived from viruses.
Okay, so in 2010 or thereabouts, an Israeli group, maybe a little bit before then, an Israeli group suggested that Israeli acute paralysis virus may partially integrate into the genome of the honeybee. In subsequent research, they didn't find it. But a paper a series of papers have just come out a couple of weeks ago, actually a couple of days ago, suggesting there may well be latent elements of bits of viral genome in many different animal species and plant species, including honeybees. So yeah, that there may be some relevance to what you say for the honeybee genome as well. Yes. Yeah. Finding those, particularly if they're not active virus, is going to be more tricky. So one needs a very good bioinformatician to work on that.
I don't want to hog the discussion, obviously, but I was really intrigued when I discovered that human beings can't even build a placenta without a piece of viral
Is that
DNA.
right? Okay,
yeah.
oh, right.
One of the layers in the placenta is viral coded. So I just wonder, it's a really fascinating subject that you're working on, Rob, because the evolutionary benefits that could be contributed to different species by the viral DNA that they're carrying is really unknown, isn't it, in the short term?
Yeah, in the case of the Israeli acute paralysis virus, the idea was that maybe by integrating some of the viral genome into the honeybee genome, the honeybee was thereby protected from that virus. So it could have a potential beneficial impact on the but I don't think it's been confirmed subsequently. But it's an interesting idea. So, and those ideas that you've put forward there, Rob, are interesting and relevant. Yes.
Really interesting. So would anyone like to ask a question now of Robert? Hold up your hand, or we'll go to the chats to see what questions people have been asking on the chats. Let's have a look.
So I can whiz. Do you want to
Yeah,
field the questions?
Well, I think you've got the questions on the chat screen, okay. Rob. So if you could take any there that you would like to answer.
Okay, so that's probably I'm just going through probably mink, right? So why no spillover in Scotland? There probably is spillover in Scotland going on, but not on places where Varroa is not present, i.e. in Colonsay, or so where there's no virus or very, very little virus. Is the spillover from honeybee, DWV from honeybee to bumblebee or vice versa? I think we've covered that, probably mainly in one direction. Let's see, we've got from Suzanne King. My question to both are social, so doesn't this increase spread within the colony? Certainly, I've not addressed viral spread within the colony. And it could be, it's needless to say, within the honeybee colony, virus can spread very easily through the colony, particularly with varroa mites, whereas that's not the case with bumblebees, probably, or to a lesser extent. From Charlie Vanden Heuvel, is any evidence that viruses are transmitted to pollen and onto bees? Yes, DVWV, for example, has been found on pollen. and it's been found in wild bee nests, in the pollen of wild bee nests. So yes, it does seem to be the case. Most likely the honeybee lands, it sucks out nectar, it collects pollen, it may regurgitate, glandular secretions may come out, it may defecate, and this can all lead to transmission of viral particles. Yeah, and certainly that means pollen can be infected and is infected. So when we run our experiments and we feed pollen, we make sure we buy bio pollen, which doesn't have pesticide, and we have to irradiate it with UV light to kill any virus. Otherwise, we could be feeding virus to them as well. Have butterflies, wasps, and hoverflies been tested? Yes, I think there are something like 70 or more different invertebrates that have been now found to be positive for deforming virus. Certainly butterflies, certainly wasps, certainly hoverflies. Yes, not all, and not at high prevalence, but they can be infected. What is the chance of transferring virus from your leather gloves and dirty bee suits? Yes, a possibility. Virus doesn't degrade so rapidly, particularly black queen cell virus. It's interesting, in the lab, black queen cell virus is a real tough virus. You can't get rid of it. It seems to be very stable. Deforming virus, a little bit less stable. We've got to put it back in the -80 freezer when we're not using it, otherwise it degrades. So yes, so possibly one could transfer it. Yes. How to get around that? I think bicarbonate of soda. Is that right? Is that what all beekeepers should have a pack of? And we've got a pack in our apiary as well, or in the bee house that we use, or the bee room, where one can wash equipment and gloves in bicarbonate of soda. Yeah, which can probably help to reduce transmission from us, by us. Thank you all. This is Kyra Wagner. Thank you. Do all of your feeding inoculation experiments use field relevant quantities of virus? Yeah, we try to use field relevant quantities. So the virus quantities that we use are thought to be then probably a little bit on the high side, 10 to the 9 virus particles that we feed in our experiments to a bee. But feeding is a very inefficient way of infecting. Injecting works very well. We can use 10 to the 2 virus particles. That's 100 virus genome equivalents, and in a pupa, and wow, 3 days later we're up to 10 to the 9, 10 or something like that. It's really, it takes off. We try to use as close as possible viral titers in our feeding experiments that reflect field relevant quantities. It's difficult to know how much they're picking up in the field though, so admittedly we might be on the more generous side. Gilly Halliday, do you share evidence with the Bumblebee Trust and equivalent? Yeah, I think I'm in contact with them, and I think they see our papers when they come out. Yes, definitely. Okay. Washing soda, thank you. Not bicarbonate of soda, sorry. There you go. Washing soda, that's what we've got. Yes, and that works well, and I think that's recommended by many beekeeping authorities around the world to sterilize, or at least to help to reduce pathogen loads of a whole range of pathogens, fungi, chalk brood, nosema and such like. It's just good practice to keep the equipment and that one's clothing then with a low titer of any pathogen. And from Ian Campbell, is there any evidence of the field density of honeybees that might impact bumblebees? Okay. So the experiment that we've undertaken in the Combi in Göttingen, we've there tried to increase densities of honeybees to see whether it might have an impact on pathogen transmission. Haven't got the data yet to know that. I've got another student working in Sardinia looking at locations with high and low honeybee densities, naturally high and low, or naturally means from beekeepers high and low, to see what impact honeybee density would have on the bumblebees. But presumably the evidence suggests, yes, when honeybee density is high, or when honeybees are concentrated in a small area of flowers, that there's probably better transmission, more efficient transmission, and therefore spillover of virus and maybe other pathogens as well. Yeah, that's the evidence that I think exists at the moment. Rob, I think I've gone through them all.
Okay.
That's probably right.
Well done then, Rob. Thank you very much.
All right. I might not have mentioned everybody's name as I answered the questions,
I'll
but
just
anyway,
have a look.
apologies for that to the people that posed the questions. Thanks for posing the questions. Thank you for listening as well.
That was really interesting, Rob. I've just remembered that the piece of DNA code in the human that codes for the placental layer that's critical to the functioning of the placenta is called SynGen1. So it's really well studied. It creates a syncytial layer with no membranes between the nuclei. So you get that transmission of nutrients across the placenta. It's quite
interesting. You know, I've just noted that down. I'll use that in my lectures to the undergraduates here in Halle once I've translated it into German. Yeah.
I think it's really important because people always see viruses as enemies, and they don't realize that when DNA gets mixed between species like this, if it serves a beneficial purpose, then it will be incorporated, which is really interesting.
It is, isn't it? It just suggests how mobile or how dynamic our genomes actually are.
Yeah.
We know there's lots of transposons that have inserted into the genome, isn't it? Something like 30% of the genome is made up of alu elements, transposons. So it's actually far more dynamic than we give credit for, I think. So it's a very interesting example there, and therefore obviously a potential origin of novelty then as well within the genome. Yeah, interesting.
I think that's it. I think the key thing is you can make a step change in your chemistry if you whip in a bit of viral code. And it'll be interesting to know if the insects have benefited as much from it as the mammals have.
I'm sure they probably have. We should probably at this point issue one of these Blue Peter cautionary words. Don't try it at home.
That's right. Well, I can't see any burning questions coming through now from people.
Some interesting comments following from your comment. That's a blue color caused by insertion of a retrovirus into the chicken genome. Very interesting.
Isn't that interesting? Yeah. Oh, well, it's a wonderful topic, Rob, and obviously it's a great importance ecologically.
I think it's important to find out how important, what impact it's having out in the field. Having seen the data, I would suggest that probably habitat destruction and degradation is #1 problem for at least for the wild bees, rather than pathogens. I'm a bit cautious there, maybe for rare species, pathogens are a problem, but probably habitat is the biggest problem for wild bees. Whereas for honeybees, it is these viruses that they're infected with, particularly deformed wing virus. I'm sure that's problem #1 associated with varroa mites. Control that and control varroa mites and the deformed wing virus. And I think we'll be back to how we used to do beekeeping in the good old days in the bee research unit down at Cardiff, yeah, but I'm trying to think of Klepper Park is the name.
Oh, Klepper Park, yes.
Yeah, where all the bees were kept from the bee research unit.
That's right.
Lovely place. Couldn't kill the bees out there in the pre-varroa days. It was very good.
I think you demonstrated very well that if the bees go to the same flowers, then there is going to be a lot of communication. And Bombus pascuorum has a longer tongue, of course, than the honeybee, and so will go to totally different flowers. Whereas Melipona beechii, where Javier was working in Mexico, that's going to go, it's a very similar tongue length to Apis mellifera, and they will go then to the same flowers, as you showed. So that was really interesting. And I think in Mayan times, Melipona beechii used to produce hallucinogenic honey because of the nectars that she was collecting from tropical flowers.
I've yet to try that.
I think that's why they had a god of the bees.
Yes.
El Dios de las abejas.
Right. Next time I go to Javier and Carlos, then I'll ask. But I suspect they keep that in there locked away somewhere.
Okay. I should hand you back now to our organizer, Harold, to complete the proceedings, Rob, but I really enjoyed that. And
thank you.
You've put me in mind. I remember from my teenage days that Martin Luther, when he was being interrogated by the Roman Catholic Church, delivered his evidence and reached a point where he simply stood and he said, "Here I stand, I can do no more." I think that's a wonderful thing to think about in that university that you're now such a key figure in. You may end up having to say that at some point to the viral world about spillover. Here I stand and I can do no more.
Or the beekeeping community. I don't know if I irate a few people, but you know, data are data, so
yeah,
they should be
You've
shared.
got to go with the evidence.
Yeah.
I'll hand you over to Harold and say thank you very much, Rob. I really enjoyed that.
No, thank you, Rob, for the introduction as well, and the kind introduction and answer and questioning at the end and complimenting very nicely. Thank you very much. Very interesting.
Thank you. Wiedersehen.
Auf Wiedersehen.
Auf Wiedersehen. Vielen Dank.
All the best, Robert.